Hi, everybody, and welcome to our module on DNA repair. In order to understand DNA repair, you have to appreciate that DNA damage occurs frequently in the life of a cell. There are many sources of potential DNA damage for a cell, including heat and UV radiation, chemicals and free radicals, and many other things. Now, this rarely leads to permanent damage of DNA material, and the reason is because numerous repair enzymes and mechanisms exist within our cells. And without these mechanisms for repair, genetic material would quickly be lost. And I just want to make a side point here. In another video, I talked about DNA mutations. A DNA mutation might be something like an adenosine nucleotide being accidentally converted into a cytosine nucleotide. The two nucleotides are still entirely normal and the genetic material is normal. It's just that the wrong base or nucleotide has been inserted to cause a mutation. Most of the things we're going to talk about in this video involve damage to the nucleotide. So it's not that one has been switched for another, it's that some actual physical damage has been done to the adenosine nucleotide, and if it's not repaired, the gene will fall apart. There are many different types of DNA damage, but two very common forms of DNA damage are depurination and deamination. Depurination actually occurs spontaneously thousands of times per day in our genes, but is repaired by mechanisms that we'll discuss in the next few slides. And depurination gets its name because it results in the loss of purine bases like guanine and adenine. At the bottom of this slide, I've shown the structures of these two bases. Deamination also occurs spontaneously many times per day. This results in a base that loses its amine group. This usually occurs in cytosine. So here's a picture to help you better understand depurination. Imagine that we have a guanosine nucleotide or an adenosine nucleotide that is part of a strand of DNA. If depurination occurs, the purine base will be lost and you will have either free adenine or free guanine. The sugar phosphate molecule will be left behind as part of the DNA strand without a purine base attached. Deamination usually occurs in cytidine. In cytidine, we have an amine group attached to the base, and if it is lost, that will create a molecule of uracil. And as you know, uracil is not normally found in our DNA. Beyond depurination and deamination, there are many other types of DNA damage that can occur when free radicals or radiation attack the rings of the bases found in nucleotides. For example, these rings can undergo oxidation or methylation or hydrolysis, and this will all damage the base found in a given nucleotide. And there are different portions of each of the bases where these processes typically occur. I'll just review a couple of them here. For example, in guanosine, methylation often happens at this nitrogen. This means a methyl group is added. Oxidative attack often happens to this double bond right here. And there are many other types of damage like this that occur at specific atoms within the rings of the different bases. So now we'll get into specific DNA repair mechanisms. There are a number of cellular systems for repairing DNA damage that have been described. They are often broken up into those that repair single-strand DNA damage and those that repair double-strand damage. And we'll talk about all of the repair mechanisms listed on this slide in this video today. The first one we'll talk about is a mechanism for repairing single-stranded DNA damage called base excision repair. This is a pathway for repairing DNA damage that recognizes specific base errors. So for example, if the base in DNA has become deaminated or oxidized, or if the ring has become open, all of these types of damage can be recognized and repaired by this system. I'll show you the prototypical example, but you should know that there are numerous variations and different enzymes used in different cells in the body, and this repair mechanism functions throughout the cell cycle in all phases of the cell cycle. To understand base excision repair, let's look at the double strand of DNA I've depicted at the bottom of the screen. And let's imagine that some damage has occurred to this thymidine nucleotide. The first step in base excision repair is to remove the damaged nucleotide and create a backbone of DNA that is missing a base in one position where there was previously a damaged base. The first type of enzyme to play a role in this process is called a DNA glycolase. There are several different enzymes used by different cells, but they all remove damaged bases. If you look at this picture of a nucleotide at the top right side of the screen here, this bond is called a glycosidic bond. Therefore, a DNA glycolase breaks that bond and lets the damaged base go free. This will create a baseless nucleotide, which is often referred to as an apurinic or apyrimidic nucleotide, meaning that it is missing either a purine or pyrimidine base. To understand the next step in base excision repair, let's look at the bottom of the screen once again. So now we have a baseless nucleotide in a single strand of DNA, And the next step in base excision repair is to remove the nucleotide backbone so that we create a gap in a single strand of DNA. There are two enzymes that play a role in this process. The first one is called AP endonuclease. This enzyme recognizes nucleotides without a base. The reason it's called AP is because the letters AP stand for apyrimic or apyrimidic, as I explained in the last slide. This enzyme attacks the 5' phosphate end of the DNA strand. I'll show you a picture of that in a minute. This nicks damaged DNA upstream of the AP site and creates a 3' hydroxyl end adjacent to the AP site. The second enzyme that can play a role in this process is called AP lyase. Some of the DNA glycolase enzymes I described in the last slide also possess AP lyase activity. This enzyme will attack the 3' hydroxyl end of the ribose sugar in contrast to endonuclease, which attacks the 5' end. To understand this better, let's look at the picture I've drawn on the screen. I've created a nucleotide that is lacking a base. This is the AP site. At the 5' end of the AP site is where you find the phosphate group here, and this is where AP endonuclease will work. It will break this phosphate bond and create a OH group here, hanging off the nucleotide above the AP site. In contrast, AP lyase will work at the 3' end. This is the 3' end of the sugar in the AP site, and AP lyase will break this bond right here. It can also open up the sugar ring, and by both of these enzymes working together, you will thus lop off this AP site and create a gap in the single strand of DNA. At this point, the final steps are fairly easy to understand. We now have a single strand of DNA with a missing nucleotide. This will be filled in a manner very similar to the way DNA is replicated if you've watched that video. DNA polymerase comes in and adds a new nucleotide that is complementary to the opposite base. Just like in DNA replication, it always adds a new nucleotide to the 3' hydroxyl terminus of another nucleotide, and then DNA ligase comes in and seals the strand, and we now have a repaired single strand of DNA where there was once damage that occurred to a base. The next DNA repair system we'll discuss is called nucleotide excision repair. And in contrast to base excision repair, this system is often used to remove bulky DNA damage that involves multiple bases, in other words, more than one nucleotide. It's often used to remove what's known as pyrimidine dimers. I'll show you a picture of this on the next page. And this type of damage is commonly caused by UV radiation, especially from sunlight. This system is active in the G1 phase prior to DNA synthesis. Basically, this system makes sure that the DNA bases are normal before the cell proceeds out of G1 to enter the S phase, which commits the cell to synthesis and division. This system is relatively simple. There are endonucleases that remove multiple damaged nucleotides, and then DNA polymerase and ligase fill in the gap. So thymidine is one of the pyrimidine bases, and two thymidine molecules can become linked together, as I've shown at the top of the screen here. This forms what's known as a pyrimidine dimer. This one I've shown on the screen specifically is called a cyclobutane pyrimidine dimer because of this structure that links the two pyrimidines together. And at the bottom of the screen is a depiction of what can happen to the double strand of DNA when this occurs. So we've got an incoming ray of light, a UV photon. This causes some of these bases to link up and form dimers, and you can see that this damages and untwists the DNA molecule. The most important thing for you to know about nucleotide excision repair for step 1 of your boards is that when this system is defective, it leads to the rare skin condition called xeroderma pigmentosum. This condition is caused by defective nucleotide excision repair in cells of the skin. Children born with this condition have extreme sensitivity to UV rays from sunlight, and that should be easy to understand based on what we just described on the last slide. This system repairs sunlight damage, so if it is dysfunctional, these children will be very sensitive to sunlight. The signs and symptoms usually appear in infancy or early childhood. These children have very easy sunburning and freckling of the skin. Their skin is dry, that's called xeroderma, that's how the condition gets its name. They also have characteristic changes in skin pigmentation, which is also how the condition gets its name. This is a picture of a child with xeroderma pigmentosum shown on the screen here. And then finally, these children are at very high risk of developing skin cancer. In fact, many of them develop various forms of skin cancer in childhood. The next DNA repair system we'll discuss is called mismatch repair. This system identifies incorrectly placed bases or nucleotides. So these are things like nucleotides that have been inserted into DNA or deleted or incorrectly matched during DNA replication. And in the video on DNA replication, I talked about how DNA polymerase can proofread DNA and fix some of its own errors. However, if that proofreading misses an error, then this system is like a backup. And this system is a little bit different from the last two we discussed, base excision repair and nucleotide excision repair, because mismatch repair is fixing errors where there's no damage to the base. The base and the nucleotide are entirely normal, so they're not going to be recognized by the base excision repair or nucleotide excision repair systems, and therefore they need this special system called mismatch repair. Also very high yield for you to know that this system is functional in the S and G2 phase after DNA synthesis. This is looking for incorrectly placed bases by DNA polymerase, so it's going to have no role in G1 before DNA has been copied. The way this system works is that the newly synthesized DNA strand is compared to the template strand, and if there are nucleotide errors, they are removed and then resealed. The mismatch repair system is very important for maintaining what's known as microsatellite stability. It turns out that the DNA in our genome has many repeating segments, and these are called microsatellites. If you look at the drawing I've made at the bottom of the screen, I've shown two complementary DNA strands as indicated by the blue and red arrows. And as I've shown them, you can see that certain segments repeat. Each blue arrow is a repeat, and there are many of these in this stretch of two complementary strands. And the repeats could be a single nucleotide, like just an A and a C, or maybe there are six nucleotides, like three A's and three C's, or maybe there are four nucleotides. But it doesn't matter. The point is that they repeat over and over again through this stretch of DNA. And you can find many sections of our genome like this. These sections are called microsatellites. In these microsatellites, a phenomenon known as DNA slippage can occur. If you look at the bottom of the screen, as I've shown, one of the repeating segments in the red strand has been pushed out. The other repeating segments of the red strand are now mispaired with the blue segments. This is called a mismatch. Luckily, we have mismatch repair systems which can fix this kind of error. And as a result, if you look at the cells in your body, you will find a stable number of repeats or microsatellites from one cell to the next. That's because mismatch repair systems are not allowing DNA slippage to modify the number of microsatellites from one cell to another cell as cells go through rounds of division. So if you understand that, it should be easy to understand that microsatellite instability is what results if mismatch repair systems are deficient. You can get what I've shown on the screen here, where you can have one cell with this number of microsatellites, and then a daughter cell with fewer satellites, and another daughter cell with more satellites, and so on. This is called microsatellite instability. It is not normal, and this is what is seen in cancer cells of some patients with colon cancer. I discussed this in detail in the GI video on colon cancer, but for our purposes here, what you should know is that if the mismatch repair system is not functioning, then microsatellite instability is what results in the cells that replicate without a stable mismatch repair system. HNPCC is hereditary non-polyposis colon cancer. It's also called Lynch syndrome, and I also discussed this in detail in the GI section in the video on colon cancer. This condition occurs in families where there is a germline mutation of DNA mismatch repair enzymes. About 90% of cases are due to mutations in genes called MLH1 and MSH2. These mutations lead to colon cancer via microsatellite instability. There's about an 80% lifetime risk of developing colon cancer if you're born with a germline mutation in one of these genes. And the hallmark of the colon cancers that develop in families who have HNPCC is that the cancer cells will demonstrate microsatellite instability. That is evidence that their mismatch repair systems have failed. I'll finish this video by discussing double-stranded DNA damage. This type of DNA damage commonly results from sources exogenous to a cell, things like ionizing radiation. It can be caused by radiation therapy, the types that are used to treat many forms of cancer. The first DNA repair system we'll discuss is called homologous end joining. So homology means a similar structure. So homologous end joining uses a similar structure, in this case a sister chromosome, as a template to repair a double-stranded break. And I've depicted this at the bottom of the screen. There's a double-stranded break in DNA. The sister chromosome is used as a template, and the double-stranded break is repaired. Contrast that with the process of non-homologous end joining. In this process, many proteins are used to rejoin the broken ends of a double strand of DNA. Some of the proteins that have been described include DNA pol lambda and mu, but there are many other enzymes that are involved as well. Importantly, this process does not use a template. That's why it's called non-homologous. And for that reason, because there's an absence of a template, it is highly error-prone. And I've depicted this process at the bottom of the screen. You have a double-strand break from ionizing radiation, for example, and then non-homologous end joining is used to bring the two strands back together. There are a couple of clinical tie-ins with double-stranded DNA repair, so let's talk about those now. The first one is Fanconi anemia, which is an inherited form of aplastic anemia that I describe in much more detail in videos in the hematology section. There are more than 13 different gene abnormalities that have been identified that can lead to Fanconi anemia, but you should be aware that many of them involve DNA repair enzymes. Children born with Fanconi anemia have DNA that's hypersensitive to DNA damage, and the cells in particular are vulnerable to cross-linking of strands of DNA. And as I said, there are many different genes that can lead to this form of anemia, but impaired homologous recombination, that's that process of double-stranded DNA repair I talked about earlier, has been described as being impaired in many cases of Fanconi anemia. The classic condition related to non-homologous end joining is ataxia telangiectasia. This condition is caused by defective non-homologous end joining systems in cells. Patients with this condition are born with mutations of the ATM gene on chromosome 11. ATM stands for ataxia telangiectasia mutated gene, and this gene is responsible for repairing double-stranded DNA breaks via non-homologous end joining. As you can imagine, if you have dysfunctional non-homologous end joining systems, the DNA is very hypersensitive to ionizing radiations in patients with this condition. As a result, they accumulate DNA damage in their central nervous system, their skin, and their immune system. I discussed the clinical features of this disorder in detail in the neurology video on the cerebellum, but I'll briefly review them here. Most children with ataxia telangiectasia are healthy for their first year or so of life. They begin walking at a normal age, but then they are slow to develop. They develop progressive motor coordination problems. That's why it's called ataxia. And by 10 years old, most of them are confined to a wheelchair. They have other symptoms related to the fact that DNA repair mechanisms are abnormal in other cells besides the central nervous system. For example, they develop recurrent sinus and respiratory infections. That's because immune system cells are accumulating DNA damage. In addition, they develop telangiectasias on their skin because skin cells are accumulating DNA damage. And importantly, there's a very high risk of cancer. And once again, this is because DNA damage occurs in cells which are exposed to ionizing radiation, and therefore cancer often results. And that concludes our video on DNA repair.